It is my pleasure to be here. Now, we are going to do this a little bit different. I used to do this thing with my oldest son, that if he never wanted to go upstairs, I had to carry him. So I'll be like, okay, I'll do one flight of stairs, and then you have to give me a kiss in the cheek. And then we will make it. So I'm going to stop every 5 minutes, and you guys have to ask a question. If not, I'm not going to keep on going, okay? That's the case. This is the title of my talk, and the way I think you guys did a great job by organizing the order of the talks for the following reason. We heard about the great studies that Manfred and Jane and also Fanny are doing, and they involve patients participating in research. And the idea is that is quite important for us to do our research. And what I'm going to tell you now is, well, what happens with like all that data? What happened with all that knowledge later on? Does it actually lead to potential therapies? And the answer to that is yes, it does. And I'm going to show you how right now. Because everything that you're going to see next comes from studies in patients that told us exactly what to do, in which directions to do to follow up our research, and then how to develop new therapies and how to read those therapies. So this is what happens when patients participate in research and go and spend a day or a week doing all these tests. That's what we did. I'm originally from Colombia, and the patients from Colombia have participated in CADASIL research for decades. And these are some of the results. So first I want to tell you that CADASIL, we always think about it as a rare disease, but there are a lot of people that have conditions similar to CADASIL. We call that cerebral small vessel disease. Believe it or not, it's the most common neurological disorder, not the most rare. It is the most common neurological disorder. It's called cerebral small vessel disease. And then a small percentage of that are people with CADASIL that have a familial form. So I just wanted to bring that to your attention, because is it rare? Well, CADASIL is, but it's a manifestation of something that is very common. Now, why do we study CADASIL? For me, this is very personal. I studied because I just fell in love with these families. Back then, when I was in medical school in Colombia, everybody was working on Alzheimer's. And I was like, there are just too many people. By the time I'm doing, I graduate and I finish all my studies, they would have a treatment for that. So I told my mentor, give me something that nobody cares about. He was like, are you sure? I was like, yeah, nobody's interested. Tell me. He was like, well, I have these families, and I think they have CADASIL. And I was like, okay, who's working on that? Nobody. And I was like, okay, give it to me. Lost causes, sign me up. So I started to work on CADASIL because of these families. And then when I came to the United States to become a scientist, then I really started to interact with the CADASIL families in the United States. And, you guys have more things in common that you have of differences. It's a great community, and I've enjoyed working with you guys. So telling you about how patients teach us things, let me tell you about this patient. So this was the case of a woman, Colombian woman. When I was in med school, I had the privilege of working with her. She was very young, 47-year-old woman. She had had a stroke. The previous stroke was at age 24. That's very early. And this is her MRI. That's the imaging of the brain of a more, like a normal individual. These are the hyperintensities that you guys are familiar with. That's abnormal. I did all the study of this family. As it turns out, we had like two very large families in Colombia. One, this woman belonged to, and then another one very large who extended that pedigree. In these patients, you know, we are asking you to participate in research. What I think about CADASIL patients is you guys are extremely generous, because we first ask you to spend like hours on end doing all these tests, then we ask you for blood, and then we are like, we may also need the brain, right? It's tremendous what we ask from patients, but it is true that all these samples are very important for us researchers. All the blood can tell us about what's happening in the brain. Then after passing, many of the patients and the relatives commit there for brain donations that then we use to do these studies and allow us to determine, for instance, what's wrong with the vessels. Those little circles up there, that's a vessel and it lights up white. That's the small muscle cells and in a control, but you see in CADASIL it doesn't light up. The small muscle cells are gone. And that's kind of like the lining of the tube that allows this tube to carry the blood to the brain, but when you have CADASIL, that essentially doesn't work very well. Now, as you know, the CADASIL is caused by mutations in NOTCH3, and NOTCH3 is actually expressed in this vessel. Like the gene makes a protein, and the protein is seen in the vessel, so it's a very strong correlation there. NOTCH3 is a receptor. It's very important for these cells to communicate with other cells, and Manfred was telling us about these cells, the small muscle cells on one side, and then the endothelial cells, much more in contact with the blood in the lumen of the vessel. Now, there is something I want you to know about CADASIL and about NOTCH. NOTCH is a very important signaling mechanism, not just in CADASIL, but there are many other conditions. All of these are diseases of the cardiovascular system, like familial tetralogy or Alagille syndrome, pulmonary hypertension, arteriovenous malformations. They all involve the NOTCH pathway. We are familiar with NOTCH3, where there is, I'm sure you figure it out, if there is a 3, where is 1, right? How about 2? Well, there is also a NOTCH4, right? So all of these receptors, it's a family of receptors, and they interact with some other proteins called the ligands, like JAG and delta 1, 2, 3. All of them are involved in the cardiovascular system. So that's important to keep in mind. And that tells us something. It tells us that NOTCH signaling is very important for the vessels, not just because CADASIL is telling us that, but because of these and many other conditions. NOTCH is also very important for other types of diseases like cancer. And NOTCH1 is very important for cancer, for instance. So this is a pathway that is very important, and that helps because that indicates that there will be a lot of interest. Maybe one drug developed for Alagille syndrome can help CADASIL. Maybe something that works for CADASIL can help cancer. And pharmaceutical companies, investors, the NIH, they are very interested in this because it optimizes resources, okay? That's like an schematic of the NOTCH receptors. It looks like a stick, sticks out of the cell. It's like a radar, right? It always sticks out, so you can actually capture the signal. That's what a receptor does. Does that make sense? Here I stop, and I'm not going to continue until I get a question. Anybody? And it could be about my work, or it could be about any of the work of the speakers, yeah? Yes, it's exactly the same. That's a great analogy. Oh yeah, the question is if these receptors work like the serotonin receptors in the brain, that essentially they are serotonin receptors, meaning they detect this neurotransmitter called serotonin. And why is it important to have receptors? Because the cell, this blob that we call the cell, has an outside and an inside, right? So things need to be communicated from the outside to the inside, and that's what receptors do. Like in that case, serotonin, or in this case, the ligands that need to contact notch receptors to communicate a signal to the cell. Sometimes, and I can tell you what that, if you are saying it's like, okay, what's the message, right? It's a signal. Tell me what the signal is. Through all my work, I think the message that the notch receptor really wants to tell the smooth muscle cell is, please survive. That is the message. And cells need that, to hear that all the time. Otherwise, they don't necessarily survive. Like, essentially, the message is, please stay. We need you here. Please do your muscle thing and stay. That is the message. And this is what I have learned. I've been working on CADASIL 25 years. This is one thing I've learned. That's the message that it wants to communicate. So when you have these mutations, these infamous cysteine mutations, that's just a type of amino acid, they affect the notch receptor. And maybe that message does not get transmitted as it should. I have taken work in my lab that I call it always to be patient-inspired. I don't necessarily want to work on things that I start from like the Petri dish and then to the mouse and go to the humans. I don't like working that way. I like working the other way around. I learn from the patients. The patients tell me which way to go. Then I test that in the mice or any other animal models. And then I test that in Petri dishes or all these very sophisticated models. And why do I do that? Well, because I'm a doctor. I like to talk to patients, but also because I think that approach may lead to better and more effective therapies. Now, what did we learn, or what did I learn from the Colombian patients? And this is true. We had a meeting just like this back in Colombia, and we invited members of the family, like the two families, and they were kind of like sitting on one side, and then the members of the other family, they were sitting on the other side. And during the, they were having breaks, they were talking to each other, and at some point a patient came in, and she was like, they have CADASIL. She came to that conclusion. That was her, her analysis of everything. Yeah, they also, this other family, they also have CADASIL, but it's different. In their family, it starts a lot earlier. I can't believe that some of them go stroke at 18. That doesn't happen in my family. That was her observation just by gossiping. Okay? And it was very accurate. We indeed validated that gossip and published it in a scientific journal. In one family, stroke started when they were much younger, in their 30s, and in her family, the one that made this observation, the stroke started much later, like 10 to 20 years much later. And as it turns out, they had different mutations. One was this C4552R, and the other one was R1031. As you can see, they are really far away. Now, as it turns out, the mutation in the patients that started a lot earlier happened to be in the region of the receptor that was critical for interaction with the ligand. It was critical for the receptor to hear the signal. So that was very important for us because it told us, hey, it could be that the mutations affect signaling of the receptor, and when they affect it a lot more, the disease is more severe. That was a critical insight. We published that ourselves in the journal Neurology back in 2002. The genotype, we call that genotype-phenotype correlation, is mutation and kind of like what you see in the patient correlation. That was validated by the European groups later in 2009. And that insight really guided all my scientific career for like the next 10 years. I was like, okay, so it seems like signaling is very important. And when the CADASIL mutation affects signaling, that makes it more severe. that was great for me because I was like, okay, let's see if that's true. And here, again, comes from the patient and the family is gossiping at the CADASIL conference. Then I go to the mice, okay? And I started to work on mice that don't have NOTCH3. If they don't have NOTCH3, I was able to study stroke in these animals. And sure enough, we found, for instance, that when the mouse doesn't have NOTCH3, they get larger strokes. And all of this white thing here, that's the stroke, and you see more white, that's bigger. So when the mouse doesn't have NOTCH3, the strokes are bigger. When they have NOTCH3, they are smaller. We're able to validate that in our animal models. Then we did all sorts of genetic tricks to validate that was true in CADASIL. So we had the, I made, I had the CADASIL families in Colombia, so I made here, but, I was here in the United States, so I was like, okay, I'm going to make mice, and each mouse would represent the family. So we made a mouse for the families with early stroke, and then we made the mouse for the family with the late stroke. I'm still working with those mice, and we give them to everybody that asks for them. So we made those mouse models, and we were able to replicate that finding of the correlation with the stroke. So let me show you, for instance, this is stroke size, and in the mice that had a mutation where that was right in the region of the receptor that was important for signaling, the strokes were much bigger. So we were able to validate that. That came from the patients gossiping at the conference. We were able to validate that result in a mouse model. Now, much later on, it was reported that as much as I can make mice that don't have NOTCH3, we also have NOTCH3 knockouts in humans. There are a few people that just don't have NOTCH3 at all. So they are like my NOTCH3 knockout mice. They're human knockouts for NOTCH3. And what we found is in these individuals, as you can see, they have, that's what we call homozygous NOTCH3 null mutation. Like, look at the images here, the MRI. You see all those white matter abnormalities. The parents, to have a kid that is a knockout, each of the parents has to lack a copy of NOTCH3, and then just by chance the kid might get it. And so the parents had like milder pathology, and the kid had stronger pathology, and they also had strokes in youth. So again, you see earlier onset, but then if you have the NOTCH3 knockout, you essentially get it in the youth. So that's very important. And again, we made even a more specialized mouse model that allows us to do something remarkable, which is have the mouse express NOTCH3 until they are adults, and then we make them knockout when they are adults. So we remove it in adulthood. Remember, that was the important message. The message that NOTCH3 tells to the cells all the time is, please survive. What if they had been hearing that all along? And then all of a sudden, in adulthood, the message stops. What we found is that if we do that in a mouse model, and NOTCH3 doesn't work now in the adult, they stop hearing the message, the cells die. Those are the gaps that you see there in the arrows. That's where the smooth muscle cells should be, and they are no longer. But they were there before. They just stop hearing the message. Now, wait a second. Okay, so Manfred, that's not a human. That's in a mouse. We see the same lesions that you see in the retina. But what I wanted to get at is that now that we know, again, patients gossiping in the conference, we validate that in the animal model. So how do we come up with a treatment? The treatment in this case, well, what if we can make a drug that sends that signal to the smooth muscle cells and tells the cells to survive? And this is the method that we developed, was to develop an antibody. The antibody is what we call an agonist. An agonist antibody is one that essentially screams to the cells, please survive. And they do it, it's actually quite a neat trick. All the CADASIL mutations are there. All of them are there. But you can actually tell Notch3 to send the message if you have the antibody binding in this region, because it essentially makes the receptor break apart and just send the signal. So the drug works as a short circuit. You've seen in many movies. The radar is still broken, the receptor is broken, but they managed to short circuit so that it still sends the signal. So we did the same trick. The antibody binds down there. It will work for the vast majority of all the mutations, because if you have been diagnosed, everybody says, oh, you have these mutations, it's in EGR repeat like 2, 3, 4, 33, 35, it doesn't matter. They are all up there. Our antibody binds here. So it will work for most of them. We tested that in cells. We have a biomarker that we can detect on the blood that indicates that it works. That's where the work of Steve and Dr. Farhelani is very important. You always need a blood biomarker. It seems like we have one. So again, that's where the biomarkers, every time they take blood, and our patients, even in Colombia, when we ask them, it's like, oh, can we have a blood sample? Well, I gave you one, like last year. This is like, what time is it? It's like, well, I don't have it. I gave it to this other person. It's like, no, it's a bad joke, but it gives the idea. We keep needing like the blood samples every time because it tells us about the status of the disease or the treatment. In this case, how biomarkers work. It's very, you can see it with this graph. Essentially, this is the levels of, let's say, Notch3 in the blood. It's actually that. And they are low, right? Then we give the antibody and the levels go up. That indicates the antibody engages the target. And that gets everybody, like investors, pharmaceutical companies, very excited because they are like, oh, this drug, not only it may work, I will know if it works. And that makes a huge difference for them. So this is a biomarker actually working. This is how they operate. We did that in cells. Now we go back to the mice that have the CADASIL mutation and we give them the therapy, and we see that it can go into the brain of the animals. Dr. Jane Paulson was telling us about like the blood-brain barrier. We have an advantage in CADASIL. It's like the weakness becomes opportunity. The blood-brain barrier is weak in CADASIL. So things like antibodies that shouldn't go into the brain actually do go into the brain. But like a lot. Talking about opportunity. So these dots are the antibody penetrating the brain. And I was like, look, it works. It goes into the brain. Why? Because the blood-brain barrier is weak. So that comes to an advantage. And we were able to show that when we give the antibody to the animal, let me see. And here is, again, where I stop, and I need a member from the audience to tell me what's different. So what's different between this vascular tree and this vascular tree? What's different? Please raise your hand and tell me. Or just scream, okay.
There's more structure to that one.
Which one?
The one on the right.
This one. More structure. What do you mean by structure?
Well, there's more branching.
More branching, right?
Yeah.
It goes here all the way to there, and then it branches, and then all the way to... So those are the smooth muscle cells. What happens here? It's less branches. It's just really like the main trunk, right? Not the branches. So this one was treated with a control. This one was treated with the Notch3 agonist antibody that we tested on the mouse because we tested it in the cells because the patients were gossiping in the CADASIL conference. Now, that's the quantification of that. And more quantification, in this case of the biomarker, working not just in cell culture, but actually working in the blood of the animals. How do you guys think I found this antibody? You think I made it? You think somebody had it? Anybody remembers how I found it? No? Okay, you guys are late in the game. Okay, so here is, maybe Barbara, this will ring a bell, because Barbara Hunt, she was our involved with the organization back in the day. So what happened was, many years ago, there was a Facebook group, and they organized the first CADASIL conference in the United States at a roadside hotel in Columbus, Ohio. And they wanted to have a speaker, and they invited me. I had just been promoted to faculty at Harvard, and they wanted me to be the speaker. And they said, well, would you come? And I said, sure. So now we took all turns. I was the one and only speaker, and I ran them through 6 hours of everything from like the genetics, the clinical, what I was doing. And at some point, I told that group what I told you guys. I said, well, notch signaling is very important. It could be very important for the vasculature. It's also important for cancer. And that's great news, because that means that there is a lot of interest, and somebody somewhere may have a drug that was developed for cancer, but it may work for CADASIL, and is somewhere stuck in the freezer. And then I saw this woman that ran out through the door. I was like, well, after 5 hours, I don't blame her. I don't blame her. She just couldn't take it anymore. And then she comes back with like a box of papers, and she runs through the stage, and she gives me a piece of paper, and she's like, you said that somebody may have a drug for CADASIL stuck in a freezer. Is this the one that you think it could work? That was that antibody. Can you believe that? Look it up on YouTube, because that's actually on YouTube when it happened, and the whole story of that conference is on YouTube. So she found it on the trunk of her car, because she's so passionate about CADASIL research. Her family had been affected, and she would just stroll the internet reading things that we never read, like patents. She found this antibody, brought it to, yeah? Correct. And I just said, with scientists, we say things sometimes rhetorically. Oh, yeah, somebody somewhere. She knew about it, and it was on the trunk of her car, and look at it now. So it's remarkable what these conferences can do and what patients can do. Sometimes it's participating in research. Sometimes it's doing their own research or gossiping, whatever works. Now, I'm going to skip through this because I want to, it's just a lot going on. I want to tell you about what we're doing now. The antibody is being developed. We set up a partnership with a company, and they gave us, the NIH gave us funding for development of the antibody, so it keeps on going. Now, what is my hope? My hope is that we keep on working, and that in 2 or 3 years from now, I call, hey, Manfred. Hi, Jane Paulson. Hi, Fanny. How's it going with those networks of patients over there? Because I may have something, and are you guys ready, or are you not? That is the hope. So this is how it goes full circle. I also call the guy in Colombia, right? Because we need all these people. We need all these people ready. We need all these networks ready. We need all the information ready so that we can actually proceed. Now, let me tell you what else I have been doing. And I have some great news to share with the community. So I also been working with Alzheimer's disease. And there are these again, families in Colombia that I told you I didn't want to work with them back in the day. Everybody knows I was too busy with CADASIL. They have a mutation that leads them to have Alzheimer's disease when they are in their 40s. So Alzheimer's usually affects people when they are in 70s or 80s, but because of this mutation, they get it in their 40s, and it's like unforgiven. If you have the mutation by age 44, they start losing memory. By age 49, they have dementia. By age 60, they are dead. No questions asked. Okay? So, and they have found in this very large family, about 1,200 people have the mutation. 1,200. And all of them, that's the history, until they found this one woman that made it all the way to her 70s, and she was fine. No cognitive impairment, no dementia, no death. She just liked to have a good life, was living independently, and she was into her 70s. So she had a mutation that predisposed her to get Alzheimer's, but we also found that she had a different mutation in another gene called ApoE that protected her. And we published that amazing discovery in 2019. That was all over the news. It was front page news in the New York Times and every other. I had people that apparently I went to like grade school with and tracked me down to tell me about this story, former colleagues. It really rocked the world because it showed us it was possible for somebody to beat the odds. And we published that in 2019. That was in Nature Medicine. It got a lot of attention. And then it really showed a lot of changes in the brain that were not what we expected for Alzheimer's. And we said, okay, well, is this one woman a one-time off, or are there more people? We actually found now recently a man, also with the mutation that predisposed him to have Alzheimer's, but he had another mutation that protected him, and we published that like a month ago, also in Nature Medicine. And again, the news took the world by storm. But now it really showed us that we had a system. It is possible for us to find people that are predisposed to have Alzheimer's, but don't get it because they have another mutation. They are protected. So here, I officially launch the Resistance to CADASIL initiative. There might be people out there, that yes, they have the mutation in NOSH3 that predispose them to develop Alzheimer's disease. But one individual may carry that mutation and may carry another one that protects them. And that would be amazing. We told the NIH about it. We wrote a grant, and we got funding to go out there and find the people that, yes, may have a mutation that predispose them to have CADASIL, but maybe they made it all the way to their 60s, 70s, or 80s, and they never got a stroke. We're looking for them. We need them, because if we find a person like that, we might be able to develop additional, what we call a protected case-inspired therapeutics. And with that, I end, and if I have time for, now I pass it on to you, because I think it's the time for questions, right? But yeah, please. For the way I do it, the question is, how many protected cases, protected individuals do I need for this research? One. So this is a different way of doing genetics, because the way we do it is, what we are looking is for extreme protection. So for extreme protection. So we're looking for 20, 30 years. Or like, oh my God, you don't even have an MRI abnormality, you are 80. Actually, back in the day, we found a patient just like that, but I was in med school, and we weren't thinking that way. We knew about this guy. He didn't want to participate in research, and he ran a farm, and eventually he agreed to see us. And he was just actually running the farm. He was 76 and working the field. So again, why is it possible to do it just with one individual? It's because we're looking with extreme protection. And yes, we look for more cases that may be protected, but that is unlikely to happen. So instead of doing that, what we do is a lot of studies, what we call molecular genetics. We essentially identify the gene candidate and make a mouse model right away and do all sorts of stem cells, like you guys are doing, Manfred at NIH. And then we test for the protection in the models, as opposed to having to identify. Yes, so the issue is here that it is known that people have a lot of mutations. How do you know it is the one? Because one individual can have like hundreds of mutations in other genes. Well, I think it's, you know, how can I say? Our approach is like a paradigm shift in genetics. The way geneticists looked at this is, okay, we only have one case that is protected. There are many candidate genes. We don't know which one it is, so let's do nothing. That's the approach, and that has been the approach. There is no p-value of statistical significance, so let's just let it be. Our approach is different. We are like, well, how about we do this research on risk? That we will waste time and resources, but maybe we find something spectacular. I can tell you, we published this in 2019. I didn't know if this was true. Oh, really, can you say that? Yes, you can, I just did. But guess what I know now? We were right on this one. It is protective. It's protective in more people. It's protected in mice. It's protected on, who was talking about cerebral organoids? Steve. It protects cerebral organoids. It's protective. So, again, talking about doing things at risk of being wrong and just going with it, I think Alzheimer's definitely needs that type of thinking. When you are like, oh, we need to think outside the box, this is what thinking outside the box looks like, okay? For CADASIL, we need that type of thinking too. We need to think outside the box, take risk, and we may have a winner. The second case, well, we just published it a month ago, so I just kind of did the same. We got a candidate, we put it out there, we did a lot of research that increased our confidence. But I don't know if we're right. We might be, and I'll tell you in 2 or 3 years. But we need to do this type of studies, because otherwise we're not going to move on. And Dr. Paulson was telling us earlier about Huntington's disease. A lot of trials going on, but Dr. Paulson, you may agree with this. That was the first gene that was discovered. Dr. Paulson, do we have a treatment? No. Okay, thank you. The first gene that was discovered for any neurological disorders for Huntington. We yet don't have a treatment. So we really need to do things much faster, much better, and maybe the standard ways of thinking don't really help. You never know who's listening, but maybe it's the baby over there, and he's the one that is going to discover the treatment for CADASIL. He was really paying attention. You never know when you give these talks, you never know who's listening and who's going to do what. Maybe he already came up with the solution. Yeah?
So this gene that protects.
Yeah?
Only found it in a couple of people. Is it, I guess, way down the line from this,
Yeah, that's a really good idea. So the question is, if for the two, one or two people that got protected because this other gene mutation, if we can then go out and make like drugs or use gene editing to do that, and then apply to other people. Yes, that is the idea. So for the grant that got approved, the first aim is the following. Well, could it be that this mutation that protects against Alzheimer's also protects against CADASIL? I mean, why not? It's protective, right? It's when you wear a waterproof jacket and then you spill yogurt on it. It's like, well, it's waterproof, right? It also, I mean, it's not that oh no, it's yogurt, I'm gonna throw away the jacket. Well, it's waterproof. What are you talking about? So it is protective. So we do hope that that's the case. Now, what are we doing? We actually, of all things, just because I have an expertise with antibody, we made an antibody against ApoE, that thing, and to see if it does the same as the mutation, and it does. Why am I saying this? Because gene therapy is out there, but I just feel like there is still a little bit of time before we can use it as treatment, but antibodies are better drugs that we can use right now. So it was just a practical consideration. But sure, if we had the technology and all the approvals to just put that mutation in people at risk of Alzheimer's, they are not gonna get it. I'm pretty sure about that one now.
And would that be with people, like adults that already have it, or when we talk about this gene therapy, is it for like people who have yet to be born? You have to.
No, it will be, so the question is if for gene therapy it will be for like people that at birth, or it will be for people that already have the disease. I would say from an ethics perspective, the way things are established right now would be for people with established disease. I'm not an expert in ethics, so I'm not gonna go into the details about at birth gene editing. I think it's too controversial, but I don't even have an informed position on that.